So this new technology we're very excited about because it can determine when cells were born in humans, and it could be, we believe it will be the modern way of looking at regeneration in humans. So until now, the only way to get an idea of whether cells were being born in humans was actually using nuclear fallout from the 1950s nuclear explosions. And that has been the state-of-the-art way of looking at cell birth in humans. So working together with Claude Lechain, who is at Brigham and Women's Hospital, we've developed an approach of using non-radioactive isotopes that can determine when cells were born. And so we've shown this in mice, and we've also shown this in humans. And the way that this is done is by using a very powerful new mass spectroscopy technique where you can image at extraordinarily high resolution variations in isotopes. And then we use stable isotopes. And so, for example, you could just drink a glass of water, and that would be completely non-toxic, and then we could come back 5 years later or even 20 years later and see what cells were born on that day. It's a very powerful way of looking at things in humans, and it required integration of clinical research and basic research and some very modern technology. So Dr. Matt Steinhauser has worked together with me and Dr. Lechain on developing this approach, and it required a lot of patience because we had to work out entirely new methods, essentially. But he has really taken this forward into humans. So the first human studies were published in Nature, and he was really the key person to really drive that.
So one way of looking at this project was that there were sort of two halves to it. One half was the method development side of that. And that was developing new approaches to tracking cells and tracking chromosomes within cells using a mass spec-based imaging platform or microscope. And we focused on developing this methodology in 3 different model systems: Drosophila, mice, and then in humans. And then the other half of the project was really taking this methodology and answering a biological question. The aspect of method development is really combining, it can be thought of as combining 2 aspects here. One is an imaging platform that provides images that are reminiscent of electron micrographs. So the imaging resolution is really spectacular. The second aspect of this methodology is that we can track different tracers, usually stable isotope-based tracers. And so really any molecule can be tagged with these stable isotopes and then administered to cells or model organisms or even humans to then track where they go in the tissues and in the cells.
So the major potential use, we think, is not only in understanding basic human regeneration, but in what happens when we use drugs and new therapies. Because the hard part about this is that we need some intermediate ways to determine whether or not we're on the right track. So just curing the disease is what we're all aiming for, but to get all the way to curing a disease is a big step. So what this will allow us to do are some intermediate steps where we can see whether or not a therapy is working. So you could test whether or not a new drug is working with a small number of patients with this technology, because we'll be able to see the regeneration actually occurring.